Hello and Assalamu alaikum. I am Ali Abbas Kazalbash, and today's program will be on the chemistry of all living cells. We see a lot of living things around us, and the first question that may come to mind is about their chemical makeup. And in that quest, we must seek the knowledge of various compounds that are involved in cellular activity. In order to understand the chemistry of a cell, we must look at the cells at the molecular level. Now, all living cells contain chemical compounds in various proportions, depending upon the structure, function, and physiological state of the cell. These chemicals are water, proteins, carbohydrates, lipids, enzymes, nucleic acids, minerals, and vitamins. Let's discuss each group one by one. Now, water is the most abundant component of all cells. The water content of an organism is related to the organism's age and, of course, metabolic activity. Water serves as a natural solvent for minerals and other substances, and water molecules also participate in many enzymatic reactions in the cell and can be formed as a result of metabolic processes. Water exists in the cell in two forms, free and bound. Now, free water represents 95% of the total cellular water and is the principal part used as a solvent for solutes and as a colloid dispersion medium. Now, bound water, which represents only 4 to 5% of the total cellular water, is loosely held to proteins by hydrogen bonds and other forces. Water acts as a dipole because of its polarity. Water can bind electrostatically to both positively and negatively charged groups of a protein molecule. Now, water is also used to eliminate substances from cell and to absorb and disperse heat. Another important property of water is its ionization into hydroxide ions, which are OH-, and the hydrogen ions, which is the H+. Our next component, of course, is proteins. Now, all proteins have the same basic structure, with a long polymer chain of amino acids, subunits linked together end to end. Now, the amino acids are basically the building blocks of proteins. An amino acid can be defined as an organic molecule that contains an amino group and a carboxylic group and a functional group designated as the R group, all bound to the center carbon known as the alpha carbon. The identity and the chemical properties of each amino acid is determined by the nature of the R group linked to the central carbon. An amino acid may have any of a variety of different R groups, often said to be the side group. When the side group is a hydrogen atom, then the amino acid is glycine, whereas if the side group or R group is a methyl group, it's said to be alanine. In addition to R groups, each amino acid, when ionized, has a positive amino or NH3+ group at one end and a negative carboxyl or COO- group at the other end, resulting in a dipolar molecule called a Zwitter-ion. Now, these two groups can undergo chemical reactions, losing a molecule or water, that's a condensation reaction, forming a covalent bond between two amino acids, resulting in a peptide bond. Now, two joined amino acid units are called a dipeptide. Condensation reactions can occur again and again, forming long chains that are typically hundreds of amino acids long. These polymer chains are called polypeptides, and each chain has an amino terminus end and a carboxyl terminus end. Now, the question that comes to mind is how these molecules are arranged in various structures of a protein molecule.
Students, we are now going to see how a protein molecule is formed. In order to understand, we must first realize that a protein is a very complex molecule. In order to understand how it is built up, we must look at its various levels of organization. These levels of organization are the primary, secondary, tertiary and quaternary levels. Let us see what is the primary structure of a protein. A protein molecule is made up of a sequence of amino acids. This sequence of amino acids is referred to as the primary structure of a protein. Whether a protein functions as an enzyme, a hormone or a structural component of a cell all depends on its primary structure. And you know, the sequence of this amino acids is so important that if I give you an example, that the human disease called the sickle cell anemia results only with the addition of a single amino acid in the protein, that is hemoglobin, which is the oxygen carrier in the blood. Now let us see what is the secondary structure of proteins. Each amino acid in a polypeptide chain interacts with its neighbors, forming weak hydrogen bonds. Due to these near-neighbor interactions, a protein molecule tends to fold into helices or coil up into wrap up into coils. This structure which a part of the polypeptide attains is referred to as the secondary structure. Therefore, we can say that the secondary structure of protein results as the interactions between the various amino acid groups present along a polypeptide chain. As you can see here, this is an amino acid chain and it has all folded up or coiled up in this case. This kind of structure is called the alpha helix. Over here you see that the polypeptide chain has folded into sheet-like pattern. For example, the protein keratin, which is found in the mammalian hair, in the fingernails and in the horns, is a structure that contains a large amount of alpha helices. Beta plated sheets is another kind of common secondary structure. Now let us see what is the tertiary structure of proteins. You see, the amino acid chains or the polypeptide chains, they further fold up. The amino acids contain side groups. These side groups, or which are also called the R groups, they interact with each other or with the backbone of the molecule or sometimes with certain structures which are found in the cell. This results in a three-dimensional folding of the polypeptide chain. The term tertiary structure refers to this three-dimensional folding of the polypeptide chain. Now we are going to see what is the quaternary structure of proteins. A protein molecule is made up of not just a single polypeptide chain, but in fact it is made up of 2 or 3 or even more than that polypeptide chains, each folded into their characteristic secondary and tertiary structures. Let me show you a picture of a molecule. Over here you can see that there are 4 polypeptide units, each folded up into their characteristic secondary and tertiary structures. This results in the quaternary structure. A very common example of the quaternary structure is the hemoglobin molecule. A hemoglobin molecule is made up of 4 heme-bearing polypeptide chains, each intertwined into a complex structure which is the hemoglobin molecule itself. Thus, the proteins that result from these various levels of organization can either be a globular protein, a fibrous protein, or a combination of the two. Do you know what are globular proteins? Well, let me tell you just a brief introduction to the globular and fibrous proteins. Globular proteins are mostly the functional proteins found in our body. For example, the protein enzymes or even the hemoglobin is a globular protein. And the fibrous proteins mostly play structural role in our body. For example, The protein keratin is a fibrous protein. Similarly, actin and myosin found in our muscles, and not to forget collagen, which is one of the most abundantly found protein in our body, are all fibrous proteins. In the end, we are going to sum up the functions that these various proteins perform in our body. Proteins are enzymes. Enzymes, they play a very important role in our body. They help to speed up specific chemical reactions that take place in the body. Proteins are also structural. For example, collagen, which is found in the tendons, bones, and ligaments of our body. Proteins can also be messengers. There are protein hormones which carry messages in the body. And proteins are also antibodies. And I'm sure that you know what are antibodies. These are the proteins that fight against diseases. And in the end, we've been mentioning hemoglobin, which is a carrier protein. So proteins also play a function to carry various substances in the body. For example, hemoglobin, it carries oxygen in the body.
Thank you, Rubaba, for explaining the various structures and functions of proteins. We shall now continue our discussion on the chemistry of life with the next macromolecule, carbohydrates. Now, carbohydrates are the most abundant form of compounds consumed daily and fulfill the basic nutritional requirements of a healthy diet, such as milk, cheese, bread, sugar, butter, cereals, etc., as illustrated here on the breakfast table. Carbohydrates are a group of molecules that contain three elements: carbon, hydrogen, and oxygen in the ratio of 1 to 2 to 1. And the empirical formula is CH2O whole n times, where the n is the number of carbon atoms. Carbohydrates are further classified as monosaccharides, disaccharides, and polysaccharides. Now, monosaccharides are the simplest of the carbohydrates and are often referred to as simple sugars. Monosaccharides taste sweet and may have as few as 3 carbon atoms, but the energy-containing monosaccharides have 6 carbon atoms. Their empirical formula is C6H12O6, or CH2O all 6 times. Sugars can exist in a straight chain form, but in solution they always form rings. The primary energy storage molecule is, of course, glucose, a 6-carbon sugar with 7 energy-storing carbon-hydrogen bonds. A disaccharide carbohydrate results when 2 monosaccharides are covalently bonded together. For example, common table sugar, which is sucrose, is made up of a glucose molecule linked with a fructose molecule. Sucrose occurs abundantly in honey and in saps of sugarcane, maple trees, and sugar beets. Sucrose is a product of a condensation reaction, in which the molecule of water is formed when 2 hydroxyl groups of the 2 sugars join together. The bond that links the 2 monosaccharides, carbon, oxygen, carbon, is called a glucosidic bond. Other important disaccharides include lactose, which are made up of galactose and glucose units, and is the predominant sugar in milk, and maltose, the sugar that sweetens malt barley, containing 2 glucose molecules. In disaccharides, the type of glucosidic bonds are alpha 1,4-glucosidic bond, as in the case of maltose, beta 1,4-glucosidic bonds, as in the case of lactose, and alpha-beta glucosidic bonds, as in the case of sucrose. Now, polysaccharides result from the condensation of many hexose monomers with the corresponding loss of water molecules. Now, their empirical formula is C6H10O5 all N times, where again N is the number of carbon atoms. Upon hydrolysis, polysaccharides yield molecules of simple sugars. Now, the most important polysaccharides in living organisms are starch in plants and glycogen in animals, which are reserve substances in plants and animals respectively, and cellulose, the most important structural element of the plant cell wall. These three substances are all polymers of glucose molecules, but differ in the way that they are joined together. The figure given here shows that the glycogen is a branched molecule in which the glucose molecules are joined together by two types of linkages. Lipids are the next macromolecules that we'll be discussing, and they are the most common constituents of the biochemical or biological membranes and are characterized by their relative insolubility in water and solubility in organic solvents. Lipids are long chains of hydrocarbons with a nonpolar hydrophobic portion and a polar hydrophilic end, rendering it capable of binding with fats and water. Lipids are esters of glycerol and fatty acids and are basically of two types, simple and compound. Now, simple lipids are alcohol esters of fatty acids, whereas compound lipids or triglycerides have three fatty acids bound to the glycerol molecule. Fatty acids have a long hydrocarbon chain with a basic or empirical formula of CH3, CH2 N times COOH, where again N is the number of carbon atoms. Now, fatty acids always have an even number of carbon atoms because they are synthesized by joining two carbon acetyl units. For example, palmitic acid has 16 carbon atoms, and stearic acid has, of course, 18 carbon atoms. Sometimes the hydrocarbon chain has double bonds between carbon atoms. Such fatty acids are known as unsaturated fatty acids. Now, the double bonds are important because they increase the flexibility of the hydrocarbons and thereby the fluidity of the biological membranes. Now, there are basically two types of lipids: natural fats and waxes. Now, natural fats are also called triglycerides. They are triesters of fatty acids and glycerol. And natural fats accumulate in adipose tissue. The carboxyl group of fatty acids reacts with the alcohol groups of glycerol in the following way, as illustrated. The repeating triglycerides, which accumulate in adipose tissue, are used by organisms for the convenient storage of energy. The oxidation state of the long hydrocarbon chains is very low, and they therefore liberate large amounts of energy, about twice as many calories per gram as carbohydrates and proteins, when oxidized to form carbon dioxide and water in cells. Now, waxes, which have a higher melting point than natural fats, are esters of fatty acids with alcohols other than glycerol, such as beeswax, earwax, or of course the wax on stoma. Waxes have a wide spread of uses, from protective coatings on fruits and leaves to protective insect secretions or repellents. Waxes protect plants from water loss and abrasive damage. Chemically, waxes are a mixture of long-chain alkanes with an odd number of carbon atoms ranging from 25 to 35 carbon atoms, and alcohols, ketones, and esters of long-chain fatty acids. The next component of our chemistry of cells are enzymes. Now, enzymes are proteins that act as biological catalysts, accelerating chemical reactions. They contain a so-called active site to which the substrate attaches itself, forming a temporary enzyme-substrate complex. Then the substrate is converted into one or more products and the enzyme becomes free again. The specificity for the substrate is considerable and frequently analogous. A slightly different shape will not work at all. The lock and key theory provides an explanation for the specificity of the active site, which is dependent upon the primary, secondary, and tertiary structures of proteins. The shape of the active site is complementary to that of the substrate. Now, according to the induce-fit theory, the enzyme-substrate interaction may cause the conformational change in the protein. The binding of the substrate to the active site is by short-range, non-covalent forces. As the enzyme-substrate complex is formed, the catalytic step in which the substrate can undergo hydration, dehydration, oxidation, reduction, or transfer of chemical groups, among other processes, proceeds. Nucleic acids, they are extremely large molecules called, of course, macromolecules. Their size results from the linking of small units which are repeated over and over again. Now, nucleic acids are found in all living things, and they are the primary factors that control the genetic or hereditary process of life. The detailed structure and function of nucleic acids will be discussed in our program on hereditary materials. Our next topic is minerals, and minerals in the living cell are found in the form of salts dissociated into anions, which are negatively charged, and cations, which are positively charged. Now, anions are negatively charged ions that accumulate on the positive electrode, the anode, whereas cations are positively charged ions which accumulate on the cathode, the negatively charged electrodes. Minerals have many important roles in cellular activity, one of which is the maintenance of osmotic pressure and the acid-base equilibrium in the cell. Retention of ions produces an increase in osmotic pressure and permits the entrance of water. Some of the inorganic ions, such as magnesium, are indispensable as cofactors in enzymatic activities. Others, such as inorganic phosphate, form adenosine triphosphate, ATP, the chief supplier of chemical energy in the living processes of cell through oxidative phosphorylation. Calcium ions are found in the circulating blood and in cells where they play an important regulatory role. In bones, they combine with phosphate and carbonate ions to form a crystalline arrangement. Now, phosphate occurs in the blood and tissue fluids as a free ion, but much of the phosphate of the body is bound in the form of phospholipids, nucleotides, phosphoproteins, and phosphorylated sugars. Now, certain mineral ions are also found as part of a larger macromolecule. For example, iron, bound to the metal-carbon linkage, is found in hemoglobin, ferritin, cytochromes, and some enzymes such as catalase and cytochrome oxidase. With this, dear students, we've come to the end of the program. I would like to thank you for your attention. Until our next meeting, wish you the very best. Khuda Hafiz.